Hello guys, welcome to this short presentation. I'm discussing viral replication, how viruses multiply themselves. First of all, why do viruses use host cells for replication? This is because viruses do not have the system that can be used to make copies of their own DNA. They do not have the ability to multiply their DNA genome and make more viruses. Okay, so they end up using the host cell systems, and these host cells could be animal cells, human cells, or plant cells. They use these cells to multiply themselves. So we have about 7 stages. The first stage is attachment, then we have penetration, uncoating, transcription, and replication, and assembly and release. So what happens in the attachment phase is that this is a stage where the virus attaches to the cell membrane of the host cell. It attaches to the surface of the cell that it wants to use for replication. When that happens, it does this because it's the only way it can release its DNA genome into this cell. Right. So attachment is simply a stage where the virus attaches to the cell membrane of the host cell that it wants to use for replication. The second stage is that of viral entry. At this stage, this is where now the virus that has attached to the surface of this cell, host cell, will inject its DNA genome or RNA genome into the cytoplasm of this host cell. Okay, so it injects its genetic genome into the cytoplasm of the host cell. Okay, in the stage 3, this is where uncoating takes place. Uncoating is simply the process by which the DNA genome or the RNA genome usually unfolds. It unfolds to expose the parts of the DNA or the parts of the RNA that must be copied. Okay, so we know there are two types of genetic genomes. There are those that are single-stranded, those that do not require uncoating, and there are those that are double-stranded. So in uncoating, it's simply a stage where the double-stranded genetic genomes unfold, exposing the parts of the DNA or RNA that must be copied. When this happens, the replication goes into stage 4, where we have transcription. Transcription is simply a process where a subcopy of the DNA or RNA genome is formed. So when these parts of the DNA that must be copied, they are exposed, the ribosomes come in, the ribosomes of the host cell, they'll come in to come and form what we call a messenger RNA in the transcription stage. This is where a messenger RNA is formed. So this messenger RNA is simply the carrier of the information that must be copied, right? So it's like a copier. It takes information to say, okay, so this is the part, this is the type of DNA that must be copied, that must be multiplied, right? So in the transcription stage also, there are certain cells, there are certain viruses that have what we call the DNA single-stranded positive sense genomes and the RNA single-stranded positive sense genomes. What that means is that during viral replication, in the transcription phase. This transcription phase will be skipped because these viruses that have positive sense genomes, they do not need to form this messenger RNA for transcription to take place. Okay, because they are already in form of a messenger RNA, for example, to just make you understand. They are already in a form of a messenger RNA, so there is no need to form another messenger RNA that can be used to make copies. So the same way this DNA genome is, it can be used to make new copies without forming a messenger RNA. So this stage is usually skipped in those viruses with positive sense genomes. In the replication phase, the fifth phase, this is where now replication takes place. Now more copies of this DNA or RNA genome are formed. So these copies from that messenger RNA that was made, they begin to interpret the information from that messenger RNA. That's the system of the host cell begins to interpret the information from the messenger RNA to form copies, new copies of the DNA or the RNA genome of the virus, because the cell system will actually think they are making copies of their own DNA genome when actually they are making copies of viruses. In the assembly phase, this is where now the DNA or the RNA genomes that have been formed, those copies of these genetic genome that have been formed, they begin to attach together to form one molecule, and they are assembled into small vacuoles. They for release into the blood, into the environment. So the DNA genomes, the RNA genomes, or the genetic genomes that have been made, those copies, they are organized and placed into vacuoles that are released in stage 7. So this release could happen through budding or rupturing of the host cell, right? So the host cell could end up rupturing and it dies completely, or it may not die because it may release these viruses by budding. I mean, budding is simply another process that have been explained under bacterial replication. Okay, this is where the cell begins to elongate in one side. Okay, on one part it begins to elongate, and then it allows that part that has elongated to cut off with the membrane and of the host cell. So that's how the viruses are released. When viruses are released in this stage, they are very small. Then they begin to mature outside and infect other cells. So this is the viral replication cycle as it happens. Thank you for listening. Please subscribe to my channel.